nuclei. Prokaryote lacks a nucleus. Eukaryote has a nucleus. Okay, the nucleus. What is the nucleus?
It's like the brain in the people center of the cell.
Who can say a bit more about it?
Okay.
Not a protein, yes.
It contains the genetic material of the cell.
Yes, it contains the genetic material of the cell, but it's what we would call an organelle. An organelle, the nucleus is a membrane. Okay, it has what's called the nuclear membrane. It contains a semi-fluid type substance known as nucleoplasm within it. The nuclear membrane contains pores called nuclear pores through which substances can leave and enter the nucleus. Who can give me, what's an example of something that might want to leave the nucleus? Yes. Proteins. More like genes, right? Genes and RNA. Okay, these are eukaryotic cells. There's the center of the nucleus. There's a structure known as the nucleolus or nucleolus. Some of you guys may have heard. What is the function of the nucleolus or the nucleolus? Who knows? The nucleolus synthesizes ribosomal RNA, also known as rRNA. What does rRNA make? What is it a part of? What structure does it help make? No, not DNA. Ribosomal RNA, no, makes the ribosome. Okay, so the ribosome consists of proteins and rRNA. Okay, and then what is the function of the ribosome?
Synthesized protein.
Synthesize proteins. And we've said bacteria and archaea are always unicellular, they're never multicellular. Eukaryotes contain both unicellular and multicellular organisms. What does unicellular mean? Single cell. What does multicellular mean? Multiple cells. As many as trillions. The shapes of prokaryotes. There are coccus or spherical. There are rods or bacilli. There are vibrio or comma-shaped. There are spirillum, which is like a rigid spiral. And there are spirochetes, which are flexible spirals. Okay, so somebody raise your hand and tell me what the shapes are and what shapes they correspond to. Who can do it? Yes,
Did they
Mary.
go to, no, I don't think I can answer that right now, but did you mean, did they go to bacteria or archaea or?
These are common shapes of prokaryotes. So these include bacteria and archaea. I mean, it's not that hard yet. You can say the name and then describe the structure that you see. Go ahead.
So the coccus is spherical.
Yes.
The rod shaped.
Well, what's another name for rod shape?
It starts with B.
Says it
I
right
can't- I
there.
know I can't see that far.
Even with glasses?
I know they don't work.
Mine.
Bacillus.
Bacillus. Okay. What about vibrio?
Yeah, I am gonna look at hers because I'm so far away. It's also rod shaped, but it's like comma shaped.
Yes, it's common shape.
And then spirillum is they're the spirals, but those are the ones that are rigid.
Yes.
And then the spirocyte are
Spirochete.
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 Spirochete are spirals that are not flaccid, but
Flexible.
flexible.
Yes. Okay. So you know that, you'll know the shapes of prokaryotes. Okay, the prokaryotic cell. Hey, grab. The prokaryotic cell. So this is the structure of a prokaryotic cell. You see, and this is the cytoplasmic membrane, also known as the plasma membrane. It's what's called the lipid bilayer. In other words, it contains lipids that are in two layers. Okay? An important point, this lipid bilayer is made up of phospholipids, which we discussed previously with regard to chapter 2. They have a hydrophilic region and a hydrophobic region. What does hydrophilic mean again? Water loving. What does hydrophobic mean? Yes. So, it has hydrophilic heads and hydrophobic tails. Now, the outside of the cell is an aqueous solution. In other words, it's watery. So, would the hydrophobic heads be oriented toward the inside of the lipid bilayer or the outside?
No.
The outside. Because on the inside, you have the cytoplasm, which is watery. The outside, you have the aqueous solution, which is also watery. So the hydrophilic heads are oriented toward the outside. Oriented toward the inside are the hydrophobic tails. Okay, where water or watery substances cannot enter. Okay, this posed a major problem for scientists for a long time. They wondered, how could water possibly go from the outside of a cell to the inside of a cell? Because it would have to pass through the hydrophobic tails of the lipid bilayer. So this was a major problem in biology. Okay? How was this problem solved? Who knows? What's that? Challenge? Channels, yes. What kind of channel?
Protein chains?
Yes. These proteins are known as aquaporins. Okay, so they're pores for water. And they are how water enters cells. The individual who discovered them, by the name of Peter Eger, he won the 2003 Nobel Prize in Medicine for discovering aquaporins. And for a long time, you could ask biologists, how does water pass the lipid bilayer? And they would have told you, I'll get back to you. And in truth, they didn't know. Okay? But we discovered the answer in the early 21st century. Ribosomes, these synthesize the proteins of a cell. They're present in every cell. The cell wall. The cell wall of bacteria and archaea. This is a structure. It's not found in animal cells. It is found in plants, in fungi, in bacteria and archaea. So do you guys remember, what molecule makes up the cell walls of bacteria? What is the major molecule? You know what you know?
Peptidoglycan?
Yes. Peptidoglycan is a major molecule found in bacterial cell walls. Okay? In plants, what major molecule is found in their cell walls? Cellulose. Yes. What about fungi? Chitin. What about archaea? Yes, say Rebecca. Pseudomurine, also known as pseudopeptidoglycan. Peptidoglycan is very important. Many of our drugs because animal cells lack peptidoglycan. Many of our drugs target peptidoglycan. Okay, penicillin, for example, interferes with the synthesis of peptidoglycan. Okay, the antibiotic. And that's one way that we're able to kill bacteria, is by interfering with peptidoglycan synthesis. The flagellum is used by bacteria for movement. The nucleoid is a region in prokaryotic cells where the DNA is. It's a name we give to something in the absence of a nucleus. These are some of the structures of prokaryotic cells. I'd recommend you learn them on your own time. Learn these structures, learn what they mean. The cytoplasmic membrane, it defines the boundary of the cell. Okay, so it determines its boundary. Historically, it's been referred to as a fluid mosaic model. In the sense, the mosaic aspect would be the proteins embedded within the plasma membrane, or the cytoplasmic membrane in this case. You have this lipid bilayer. These are what I mentioned are the hydrophilic heads right here. And these are water-loving. Oriented within them are hydrophobic tails, oriented this way and oriented this way. So these are the hydrophobic tails of the phospholipid. There are proteins associated with the cytoplasmic membrane. Some of them are integral proteins. They span both sides of the membrane. Some of them are peripheral proteins. They're just on one side of the membrane. These are very important for cell communication, moving things in and out of the cell. Extremely important. In eukaryotic cells, there's a protein, a type of protein, a class of protein, known as G protein coupled receptors, or GPCRs. About 35% of our current drugs approved by the FDA target G protein coupled receptors. Okay? Over one third of our drugs target G protein coupled receptors. You can imagine then that they're extremely important. Indeed, the people who first discovered G protein coupled receptors back in the 80s or 70s, they went on to win the Nobel Prize in Chemistry for doing so, because they're such important pharmaceutical targets. The cytoplasmic membrane is often referred to as selectively permeable. There are some substances that can simply move across the membrane. Okay, other substances are either too large or too polar or something of that nature. They cannot move across the membrane. They require proteins, the help of proteins. So for example, oxygen, CO2, nitrogen, and water pass freely. Okay, they can have proteins, permeases that allow them to pass, but they do pass freely. Some cells facilitate water passage with aquaporins. Other molecules must be moved across the membrane via transport systems that allow them to move across the membrane. For example, sugars, ions, amino acids, ATP, and macromolecules cannot pass through the membrane, whereas oxygen, CO2, nitrogen, these can move across the membrane. Okay. So, simple diffusion. Simple diffusion is when molecules move from an area of high concentration to an area of low concentration. For example, let's say I had a bucket of water, okay, and I took a drop of food coloring, say red food coloring, and I put a drop of it in the water. What would happen to the food coloring in the water? It would at first be highly concentrated, right?
Yeah.
But over time, the molecules would spread out throughout the water. So they're moving from an area of high concentration to an area of low concentration. Make sense? And that happens passively. In other words, you don't need to use energy to make it happen. It just naturally happens. But to move things from an area of low concentration to an area of high concentration, that does require energy. Let me give you guys an example. When you guys inhale, you take in oxygen. The oxygen in your lungs has a low concentration. So, oxygen moves into your lungs where it has a low concentration. And, it goes into your lungs, it moves into your blood where the oxygen has a low concentration. Carbon dioxide, which is highly concentrated in your blood, leaves your blood, goes into your lungs. Make sense? So, oxygen in your lungs goes into your blood. Carbon dioxide in your blood goes into your lungs. And then you exhale, you remove the carbon dioxide from your lungs, and then when you breathe again, you take in more oxygen. That is how your blood becomes oxygenated. Okay? Now, to yap a bit more about this. Your cardiovascular system has two circuits, okay? The systemic circuit and the pulmonary circuit. Which one is which, and what does it do? Who knows? What is your pulmonary circuit and your systemic circuit? What do they do? Yes?
Systemic circuit sends deoxygenated blood back up, and then your pulmonary circuit sends out oxygenated blood.
Not quite, not quite. Who else thinks they know? Yes?
Pulmonary is like the deoxygenated blood, and the systematic is like it's the oxygen it's carrying when you breathe, like the oxygen to your blood, like to nutrient, like tissues.
So, the pulmonary circulation sends blood to your lungs. The purpose of the pulmonary circulation is to pick up oxygen. Your systemic circulation sends oxygen from your blood to all the tissues in your body. Okay? So let's say, who owns a business? Does anybody own a business?
Dad does.
What's that?
My dad does.
Oh, what does your dad do?
He's a mechanic.
Okay, so he's a mechanic. Before he can do his job, does he have to pick up tools? Or like stuff for the job?
No.
No? Okay, maybe not. What about, does anybody have like an electrician or something like that in their family?
My boyfriend's family owns their business for construction, and
Yeah.
they always go to Home Depot first
Yes.
and pick up everything they need for the day,
Yes.
and then use it.
So, the point is, you need to go to the lungs and pick up oxygen. There's no point going throughout the body if you don't have oxygen to give to the cells. So before you can do your job, you have to pick up your equipment. And you pick up your equipment at the lungs. Okay? So your body, let's say it's full of oxygen, all your blood. It's gonna go around to the cells, it's gonna give them all oxygen. Okay, then it's gonna go back to the heart, but it can't go in the systemic circulation again, because by the time it's given oxygen to all the bodies in the cell, or all the cells in the body, it's gonna be low on oxygen. So it needs to go through the pulmonary circulation, pick up more oxygen, and then it can go around the systemic circulation again. Does that make sense? So you go to the pulmonary circulation, you pick up oxygen, you come back to the heart, you go in the systemic circulation, you give that oxygen to all the cells of the body, you come back to the heart, you pick up more oxygen, you do it again. Come back to the heart, pick up more oxygen, do it again. Make sense? The heart has 4 chambers. What are those 4 chambers? Yes?
Your
ventricles? Right and left ventricle?
I can't remember the.
Yes,
Your atria.
yes. 2 ventricles, 2 atria. On your left side, these are known as the left atrium and left ventricle. On your right side is the right atrium and right ventricle. Okay? Your atria receive blood from the circulation. Okay? So, your ventricles send blood into the circuits. So your right ventricle, does that send blood into your systemic circuit or your pulmonary circuit? Pulmonary circuit. Your left ventricle sends blood into your systemic circulation. Now, in which case does the blood have to travel farther? In the systemic circulation or the pulmonary circulation? Systemic, because the lungs aren't that far away. As a result, the left ventricle is thicker and larger than the right ventricle. Make sense? Can anybody summarize what I've said so far? Anybody want to try? Yes, go ahead.
So you're saying that blood starts, let's say, in the left atria, goes to the left ventricle, then goes to the pulmonary.
No.
So it's deoxygenated blood.
It doesn't go to the pulmonary from the left ventricle. It goes into the systemic from the left ventricle.
To the right, from the right ventricle to the pulmonary.
Yes, pulmonary, yep.
Then it goes back to the heart, to the right atrium, or left
Left atrium.
left ventricle to the systemic.
Yep.
To put out oxygenated blood.
Yes. Yeah, that's about it. Which one's thicker, the left or the right ventricle? Left. Okay? So, it depends where you start in the circuit. But depending on where you start, it's easy to figure out what happens next. Okay? Blood is always received by the atria. Okay? Blood is always received by the atria. And from the atria, it goes into the ventricles. If it's received by the right atrium, it'll go to the right ventricle. If it's received by the left atrium, it'll go into the left ventricle. Make sense? Okay. If your blood is going into your systemic circulation, what blood vessel will it pass first? What is the largest blood vessel in the body? The aorta. So, if it's going to go in your systemic circulation, from your left ventricle, it's going to go into the aorta. If it's going to go into your pulmonary circulation, what is it going to go through?
Lungs.
What's that? Not the pulmonary veins. Pulmonary arteries, from the pulmonary trunk into the pulmonary arteries. Okay? Arteries always take blood away from the heart. Veins always take blood back to the heart. You might have this idea that arteries are always oxygen rich and veins are always oxygen poor. But that's only true for the systemic circulation. In the pulmonary circulation, veins are oxygen rich, arteries are oxygen poor. Okay, so when we discuss pathogens that target the cardiovascular system way ahead at Unit 4, you guys will have some idea of what I'm talking about. And I'll review with you guys too. So simple diffusion. Molecules go from an area of high concentration to an area of low concentration. Osmosis is the diffusion of water. So when water goes from an area of high concentration to an area of low concentration. Now, when would diffusion occur? It often occurs when the solute is impermeable, when the solute cannot pass the membrane. Okay, so normally we're told that solutes go from an area of high concentration to an area of low concentration. But you might imagine if we had a membrane that, let's say a very large molecule like starch, couldn't pass through the tiny pores in the membrane, then it wouldn't be the solute that would go across the membrane, it would be water. Okay, because the membrane is impermeable to a particular solute. So, in a hypotonic solution, what is that? What's that?
Low water concentration?
Well,
Or water,
For soda?
isn't it where water goes up, like, it goes in hypoatomic and then hyper is when it exits?
so, in a hypotonic solution, the solution outside of the cell is such low concentration, it's so dilute that water enters the cell. Okay? Living organisms that have a cell wall, they tend to prefer hypotonic solutions. Okay? Where water enters the cell. Plants, for example, prefer a hypotonic environment. In fact, if they're in a situation where there's not enough water outside of their cells, they will shrivel up and die, as you've probably noticed if you haven't watered your plants. Okay? Right. So in a hypertonic solution, the solution in which the cell is in is so concentrated that water leaves the cell and goes out into the environment. Okay, when this happens, for example, in plant cells, a phenomenon known as plasmolysis occurs, where the cytoplasmic membrane detaches from the cell wall. Okay? There's a situation where there's equal concentrations of solution between the cell and the solution in which it's in. That is known as isotonic. Animal cells prefer an isotonic solution. Bacteria, plants, they prefer a hypotonic solution. If your red blood cells, for example, are placed in a hypotonic solution, a situation known as hemolysis will occur. The cells will burst, okay? The cells will lyse, as it's called, and the cells will die. Some of you guys are going to be nurses. Do you guys think it would be a good idea to give a patient a solution of pure water?
No.
An IV of pure water? Probably not, right? Because the solution is hypotonic to red blood cells, and the patient will, their red blood cells will burst and die if that happens. Okay, so very dangerous situation. That's why you never give pure water in an IV. You give a solution that's about 0.7% sodium chloride, so that the solution remains isotonic with the red blood cells. The environment of prokaryotes is typically dilute. Water flows into the cell, where cytoplasm is a concentrated solution. The cell wall, just like in plant cells, prevents the cell from bursting. Most molecules must pass through proteins functioning as selective gates. These include transport systems, which are permeases or carriers. They span the full membrane. So the molecule binds to a part on the protein, the protein alters its shape and it allows the molecule to leave the outside of the cell and enter the inside of the cell. How do you guys think most drugs get into a cell? What do you guys think? Well, they happen through simple diffusion. Okay, so the molecule just diffuses into the particular place where it's supposed to be. There's a major problem in medicine and biology. It's known as the drug delivery. So we can often develop drugs that will treat a particular condition. The major problem is how do we get those drugs to where they need to be? Because many such drugs are degraded, they're taken up by other cells. There's no way to get what a particular drug that we need to get somewhere, to get it to that place. So many scientists are working on this problem. It's known as the drug delivery problem. So, it's various methods have been tried. One of the most important people involved in this field. Let me put my phone on silent. One of the most important people in this field is a chemical engineer by the name of Dr. Robert Langer. Okay? Robert Langer, he graduated with what would be the equivalent of a PhD in chemical engineering from MIT in the 1970s. He subsequently went on to complete a postdoc at Boston Children's Hospital, working under a scientist named Judah Folkman. Judah Folkman hypothesized in the 1970s that cancer required a condition known as sustained angiogenesis. What does angio refer to? Blood vessels. What does genesis refer to? Creation. So cancer requires the creation of new blood vessels because these blood vessels must nourish a tumor. When Judah Folkman first proposed this hypothesis, many scientists were skeptical. They didn't believe that new blood vessel formation was required for cancer, for the metastasis of cancer, the movement of a tumor to distal sites in the body. So he put Robert Langer, his new postdoc, on this job, and he checked hundreds of different molecules. What substance in the body do you guys know of that's free of blood vessels? Cartilage. Cartilage lacks blood vessels. So, Langer assayed hundreds of molecules found in cartilage until he found his angiogenesis inhibitor. Okay? And today we can use these drugs, these angiogenesis inhibitors, to treat people who have cancer. By cutting off the formation of new blood vessels, we can essentially starve the tumors to death. That also opens up other possibilities. One of my hypotheses is that to treat people who have clotted arteries, we might use angiogenesis promoters. For example, myocardial infarction or heart attack is often associated with a clottage of the coronary artery. Well, what if we use angiogenesis promoters to create new arteries in the heart and therefore develop a way to bypass blocked arteries for the heart? Could be a way to save people's lives. What I was referring to about the drug delivery problem, Langer has done a lot of work for the drug delivery problem. There are toxins that are produced by microorganisms. For example, Clostridium tetani is the bacterium that produces the toxin tetanospasmin. What do you think tetanospasmin causes? Tetanus, right? Clostridium botulinum produces the botulinum toxin. What disease do you think it causes? Botulism. Vibrio cholerae produces the cholera toxin. It causes? What's that? Cholera, yes. Now, all of these exotoxins, as they're called, are known as AB toxins. So they're proteins. They have an A subunit and a B subunit. The purpose of the B subunit is to integrate into the host cell plasma membrane. Once the B subunit is in the host cell plasma membrane, it'll shuttle the A subunit into the cell. And the A subunit will exert the toxic effects. Okay, so cholera toxin, that's an AB toxin. Tetanus, tetanospasmin is an AB toxin. Scientists are now hijacking the B subunit of various toxins. The reason they're hijacking it is because they can use the B subunit, they can attach it to something that's beneficial, and then the B subunit will deliver the beneficial thing into the cell. So this is a way of bypassing the drug delivery problem by using the B subunit of AB toxins to deliver a particular molecule in a cell. Make sense? Facilitated diffusion is a form of passive transport. Pay attention, guys. Facilitated diffusion. You use a protein to carry out diffusion. So you're still moving molecules from an area of high concentration to an area of low concentration. Active transport. This is transport that requires energy. Okay, so you use a pump. You move something from an area of what concentration to an area of what concentration?
Low to high.
Low to high. Low concentration to high concentration. Okay? And this requires energy. What is the molecule that serves as the energy currency for life?
ATP.
ATP. So it requires ATP. An example is an efflux pump. An efflux pump moves molecules out of the cell. For a bacterium, what kind of molecules do you think it might want to move outside of its cell?
Right. Wait, no.
Well, antibiotics, for example. A bacterial cell might want to save its life by moving antibiotics out of the cell. So here are the transport mechanisms. You can look over these. The cell wall. So, there are two types of bacterial cells. There are gram-positive cells and gram-negative cells. Gram-positive cells have, outside of their cytoplasmic membrane, a thick layer of peptidoglycan. This can be as many as 30 sheets thick of peptidoglycan. Peptidoglycan consists of two types of molecules. One of them is N-acetylglucosamine. The other is N-acetylmuramic acid. These can be called NAG and NAM, and they alternate. You go NAM, NAG, NAM, NAG. Who can repeat those longer names for me? N-acetylglucosamine and N-acetylmuramic acid.
Can you say the second one again, please?
N-acetylmuramic acid. Okay. So, gram-positive cells have a thick layer of peptidoglycan outside of their cell. Gram-negative cells have their cytoplasmic membrane, a thin layer of peptidoglycan, and then an outer membrane. And the space between the two membranes is known as the periplasm, or the periplasmic space. Examples of gram-positive cells are bacillus, such as Bacillus anthracis, the causative agent of anthrax, Staphylococcus, and Streptococcus. Gram-negative cells include Escherichia, Neisseria, and Pseudomonas. Neisseria gonorrhoeae causes the disease gonorrhea. E. coli, there are strains of E. coli that possess Shiga toxin, which causes a very severe disease. There are also strains of E. coli that harbor enterotoxin, which causes a very severe diarrheal disease characteristic of cholera. Yes.
Can I take layer test it over again? Does that make gram-positive, like a stronger type of bacteria because that layer is so thick?
No,
Okay.
no. They have different properties. Yeah, they have different properties, but I think that's a simplistic way of thinking about it. Teichoic acids are present in gram-positive cells. So know that. Teichoic acids are present in gram-positive cells. What's that?
What's an acid?
Teichoic acids.
Please spell it, sorry.
Teichoic. Now, gram-negative cells. They also have structure found in their cells known as LPS, or lipopolysaccharide. Write that down. Lipopolysaccharide. This means it consists of lipid and polysaccharide. A component of lipopolysaccharide, or LPS, is known as lipid A. So, lipopolysaccharide is recognized by the immune system. If it's present locally in a particular area only, it can help speed up recovery from damage. However, lipopolysaccharide, and specifically that component of it, lipid A, if it gets into the bloodstream, it can trigger a systemic response, systemic inflammation, and it can cause sepsis, okay, which is a systemic response to antigen. We haven't discussed what an antigen is yet. This systemic inflammation can be very dangerous, okay? It can lead to septic shock, which is characterized by very low blood pressure. And by the way, low blood pressure is more dangerous than high blood pressure. So blood cannot move through the arteries. And when blood can't move through the arteries, you risk circulatory collapse, dying due to blood clots, because your blood is unable to move through the blood vessels. So lipopolysaccharide, very dangerous. It's only present in gram-negative bacteria. And by the way, it is not destroyed by heat. So you cannot destroy endotoxin, which is lipopolysaccharide, with heat. It's another name for LPS, by the way, is endotoxin. It's only found in gram-negative bacteria. And the lipid A component of lipopolysaccharide is very dangerous. If it gets into the bloodstream, it can lead to septic shock, which is a very low blood pressure, and it's not killed by heat. By the way, there are vaccines known as toxoid vaccines. Toxoid vaccines are inactivated toxins. Okay, so you take bacteria, some of the bacteria I've mentioned, and you inactivate their toxins through heat, for example, and then you can inject people with them and they serve as a vaccine against the actual disease. With endotoxin, we cannot create toxoid vaccines. Okay, so very serious. There was a danger of, in the past, giving people blood that contained LPS, lipopolysaccharide, or endotoxin. There's severe danger, and people have died because of that. They received blood with LPS. A test has been developed, known as the limulus amoebocyte lysate assay. Yes. No, LAL. LAL test. So basically, you take blood from the horseshoe crab and you introduce the substance into it. And if you have only a tiny amount of endotoxin, the blood will form a gel. And that allows us to know if the blood contains endotoxin. It's known as the limulus amoebocyte lysate assay. LAL. So endotoxin, very dangerous. There are two types of toxins. Exotoxin, I've given you guys one example of exotoxin. These are the AB toxins. And I've just explained to you endotoxins. And we'll discuss endotoxins more later in this class. So, what is endotoxin? LPS, which stands for what? What's that, Yuli?
The lipopolysaccharides.
Lipopolysaccharide. What component of LPS or lipopolysaccharide is specifically dangerous? Lipid A. Okay, and what can happen if lipid A gets into the bloodstream? Sepsis and septic shock. Okay, you get a systemic inflammatory reaction that can lead to a life-threatening drop in blood pressure. What test do we use to detect endotoxin? LAL, limulus amoebocyte lysate assay. And what is it? What is the test? What is it derived from? Which organism? The blood of which organism? The horseshoe crab.
And you said that when a little bit of that LPS is introduced to the horseshoe crab, it's only a tiny amount, you can coagulate?
We take the blood from the horseshoe crab, we introduce a substance we want to know if LPS is present in, and even a tiny amount of LPS can be detected using the LAL assay. Okay, another question, endotoxin, can it be destroyed by heat?
No.
No, it cannot. Okay, many exotoxins, they can be destroyed by heat. Endotoxins cannot. Okay, so, the peptidoglycan, N-acetylglucosamine, N-acetylmuramic acid, N-acetylglucosamine, N-acetylmuramic acid. There's a tetrapeptide, 4 amino acids here and here, and these are connected through a peptide interbridge that connect the sheets of peptidoglycan. Okay, so it's a three-dimensional structure. Many or several antibiotics target this part of the interbridge. Okay, they prevent the sheets from linking to one another, cross-linking, which disrupts the peptidoglycan structure and kills the bacterial cell. Do humans have to worry about molecules that target peptidoglycan? We do not. And that's why these drugs work on us, because they don't harm us directly, but they do harm the organisms directly. However, they can lead to dysbiosis, disruption of our microbiome. So that's something you need to worry about. More three-dimensional structure. So here's the cytoplasmic membrane. You can think of the peptidoglycan as a three-dimensional structure. And in gram-positive cells, it's quite thick. That is outside of the cell. Gram-negative cell wall. You have the cytoplasmic membrane. You have a thin sheet of peptidoglycan. You have an outer membrane. By the way, this is known as a periplasm between the membranes. And then out of the membrane, you have this very dangerous endotoxin, or lipopolysaccharide, which contains lipid A, which is the endotoxin. Outer membrane is a bilayer made of lipopolysaccharide. Here are some bacteria that might be termed pleomorphic. They have multiple shapes. So these bacteria, some of them lack a cell wall, although it's less common. Capsules and slime layers. I want us to get out of here pretty soon, so I won't. It's okay. We've only got 5 more seconds. So you guys remember, I told you guys about Frederick Griffith's test with mice, right? You guys remember? He was the one who first discovered the transforming principle, right? Which was later determined to be the genetic material, or DNA. Remember, he worked with 2 strains of bacteria. One was an R strain, or rough strain. The other was an S strain. What do you think the difference between them was, the R strain and the S strain?
One's rough and smooth.
Yeah, one's rough, one's smooth, but why is one rough and one smooth?
Rigid cell wall.
One of them has a capsule. The S strain had a capsule. And that capsule is important for evading the immune system of the host. Actually, guys, we'll be back at 7:05. I need to use the bathroom and things like that. So, come back 7:05.
My son's 5.
I heard 12 and 15 and then 12 again.
I thought it was 5.
Really close.
So how do you say space?
Got me off of two. Just give it to me, I'll do it. You tell me how to do
Because the electrons are not even positive. We're just supposed to memorize it.
Really good. I'm trying to figure out
It's like
I was just like, you know what, I don't know where she is.
See, I just remember these 2 because this is basically double except for this one. Because he said it wasn't permanent. You were asking me to check this one. This one's and you sort of like this. No.
Yeah, I have all this right here.
I'm missing it. I had one. I can see this.
It's weird because this is 5, it takes 3 to make 8. So I would know it makes these bonds, you know, then that would be easier.
I have a check and free download for you without it. All the slots, we look at it in.
Everyone's had one protein. there's just so much to because I'm training to you that I know nothing about the industry. It's all on me for today because it's only a super job.
Polymers. Capsules, they help prokaryote by allowing a bacteria to evade the immune system. Okay, so it allows it to not be destroyed by the immune system. One of the reasons it does that is because the structure of a capsule is in such a way that it's difficult to produce antibodies that can target it. So pillae, these are often used for attachment. They're shorter than flagella, but they're present on bacterial cells. So you can see these hair-like structures. Chromosomes. Bacteria usually have a single chromosome, one single circular chromosome. Plasmids are extrachromosomal, so they're not chromosomes. They're circular structures that can divide, that they can replicate without being part of the chromosome. So you can produce a copy of plasmids. Bacteria can actually pass on their own plasmids to other bacteria. Many plasmids contain genes that are involved in, that produce proteins involved in antibiotic resistance. So some plasmids contain genes for resistance against the antibiotic tetracycline. These are plasmids. Ribosomes are involved in protein synthesis. The cytoskeleton of a bacterial cell is a structure near the plasma membrane that consists of proteins. They allow the bacterial cell to alter its shape and move substances across it. Storage granules and vesicles, you can see here storage granules. They store molecules important for the survival of the bacteria. Endospores. So these are important. Endospores are produced, or spores, are bacterial structures that allow the bacteria to survive very severe conditions. Okay? The genera, Bacillus and Clostridium, are very important endospore formers. Okay? You guys remember when I told you about the anthrax mailings in 2001, right? The individual who mailed those letters, he took Bacillus anthracis that were endospores. Okay, and once the letters were opened, or when they were mailed, they began to germinate. They acquired their active living state. So they weren't dormant anymore, and that's how they killed those postal workers and members of the media, etc. Endospores, extremely resistant. They may remain dormant for a century or longer. They're extremely resistant to heat, drying out, chemicals, UV light, boiling water. So endospores that survive can germinate to become vegetative soil. They're found virtually everywhere. Remember, use this to learn the structures especially. I want you guys to know the structures of a bacterial cell. And with that, put everything off your desk, David,